Khateeb series in Human Anatomy and Physiology. Chapter 2, Cell Structure and Function. Episode 4, Cell Organelles and Function Part 1. Cell organelles are specialized structures within a cell, each with a unique role essential for the cell's survival and function. To understand these organelles, let's first look at the cytoplasm, which is the gel-like substance that fills the space between the cell membrane and the nucleus. The cytoplasm consists of a fluid portion containing dissolved substances called the cytosol, as well as from various organelles, and a supportive network of cytoskeletal proteins that help maintain the cell's shape and facilitate movement. Now, cell organelles can be categorized into two types: membranous and non-membranous. Membranous organelles are enclosed by one or more lipid membranes, which create compartments within the cell to carry out specialized functions. These organelles are found primarily in eukaryotic cells. Some key examples include the nucleus, mitochondria, rough and smooth endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. On the other hand, non-membranous organelles do not have a surrounding membrane and are present in both prokaryotic and eukaryotic cells. These structures play essential roles in providing structural support, protein synthesis, and cell division. Examples of non-membranous organelles include ribosomes, cytoskeletal proteins, centrosome, centrioles, and proteasomes. Together, these organelles work in harmony to ensure the cell functions efficiently, allowing it to grow, reproduce, and respond to its environment. In this session, we are going to discuss the structure and function of these organelles in order. Deep within the heart of every eukaryotic cell lies the nucleus, the largest organelle in the cell that serves as the cell's command center as it stores our genetic material, the DNA. Enclosed by a double membrane known as the nuclear envelope, the nucleus safeguards the genetic material, isolating it from the cytoplasm while allowing selective exchange through nuclear pores. These microscopic gateways regulate the movement of molecules, ensuring that essential instructions flow seamlessly between the nucleus and the rest of the cell. Within the nucleus resides the chromatin, which carrying our genetic DNA, essential for growth, development, and reproduction. Let's magnify the cell chromatin to understand its structure. As shown in this image, the chromatin is a non-condensed complex of DNA and protein that exists in non-dividing cells. The proteins are bound to DNA are called the histones and are crucial for packing the long strand of DNA into the nucleus. However, these repeated DNA protein subunits forming the chromatin are called the nucleosomes. Therefore, when the cell is not dividing, each chromosome is composed of a single chromatin fiber. However, during DNA replication and cell division, each chromatin condenses to form a chromosome that is composed from two sister chromatids. After cell division, sister chromatids are distributed in the daughter cells, and each chromosome consists of one chromatid. Now let's understand the role of the nucleus as a control center of the cell. In this regards, the nucleus governs cellular activities by controlling gene expression through the process of transcription, where DNA is transcribed into messenger RNA or known as mRNA. This mRNA then travels to the cytoplasm to guide protein synthesis at the ribosomes in a process called translation. At the heart of the nucleus lies the nucleolus. The nucleolus has a dense structure and contains DNA that is responsible for synthesizing ribosomal RNA and assembling ribosomal subunits. However, as we can see in this image, the outer nuclear membrane is continuous with the rough endoplasmic reticulum, which contains the ribosomes needed for the translation process or protein synthesis. Finally, with respect to number of nuclei, many cells in our body have only one nucleus, such as epithelial cells and smooth muscles. Other cells are multinucleated containing more than one nucleus. A good example is a skeletal muscle cell. However, there are also some cells that lack nuclei or setonucleated, such as red blood cells. Now, let's move on to the next organelle in our discussion. The mitochondrion, which often referred to as the powerhouse of the cell. This image illustrates the structure of a typical eukaryotic mitochondrion. The major function of the mitochondrion is the site where the aerobic phases of cellular respiration take place, producing energy in the form of ATP. More specifically, this is where the Krebs cycle and the electron transport chain occur. However, it's important to remember that glycolysis, the initial phase of cellular respiration, happens in the cytoplasm before the products enter the mitochondrion. Now, let's analyze the structure of the mitochondrion. As shown here, each mitochondrion is a double-membraned organelle, meaning it has both an outer membrane and an inner membrane. The space between these membranes is known as the intermembrane space. However, the outer membrane is smooth and acts as a protective barrier. It contains porins, which are channel-forming proteins that allow small molecules, ions, and nutrients to pass freely between the cytoplasm and the inner membrane space. The inner membrane, on the other hand, is highly specialized. Unlike the outer membrane, the inner membrane does not have porins, making it highly selective about what can pass through. This inner membrane is also folded into structures called cristae. This cristae significantly increase the surface area, providing more space for proteins and enzymes involved in ATP production. Importantly, the inner membrane contains essential proteins and ATPase enzyme required for the electron transport chain. So the inner membrane is the location where electron transport chain happens. Inside the inner membrane, we find the mitochondrial matrix, a dense, gel-like substance that contains enzymes, ribosomes, and mitochondrial DNA. This matrix is where critical metabolic reactions occur, including the Krebs cycle, which generates molecules necessary for ATP synthesis. Meanwhile, the inner membrane space plays a crucial role in cellular respiration by storing protons that are pumped out of the matrix during the electron transport chain. From this discussion, we can recognize that one fascinating feature of mitochondria is that they have their own DNA and ribosomes. This means that the mitochondria can produce some of their own proteins and even replicate independently of the cell. Imagine a world where nothing grows, nothing heals, and life simply cannot exist. Sounds impossible, right? Well, that's exactly what would happen if cells lacked one of their most powerful and essential machines that is known as ribosomes. These tiny structures are the master builders of life, responsible for creating the proteins that fuel everything from your muscles to your brain. Without them, life as we know it wouldn't be possible. Now, let's take a closer look at ribosome structure and function. But don't be fooled by their size. Ribosomes may be microscopic, but they are incredibly sophisticated. Each ribosome consists of two interlocking subunits, one large and one small. These two subunits function like a precision-engineered machine, synthesizing proteins from messenger RNA in a process called translation. This process also requires another RNA molecule known as transfer RNA, which contains an anti-codon trinucleotide sequence that is complementary to a corresponding codon in the messenger RNA. This transfer RNA also carries the matching amino acid. We will cover the complete process of translation in other episodes of this series. However, what exactly makes up a ribosome? Ribosomes consist of approximately 60% ribosomal RNA and 40% ribosomal proteins. The ribosomal RNA is a type of non-coding RNA that acts as a ribozyme or an enzyme like RNA to facilitate protein synthesis. This ribosomal RNA is transcribed from ribosomal DNA within the genome, which then binds to ribosomal proteins to form the small and large ribosomal subunits. Next, do you think there is a difference in prokaryotic and eukaryotic ribosomes? In fact, ribosomes are present in both prokaryotic and eukaryotic cells. But they differ in size, structure, and composition. Prokaryotic ribosomes, found in bacteria and archaea, are smaller, measuring 70 Svedbergs, are known as 70S. The Svedberg unit, abbreviated by the letter S, is a measure of how fast molecules sediment during centrifugation. As shown in this image, the 70S ribosome in prokaryotes consists of two subunits: a 50S large subunit and a 30S small subunit. On the other hand, eukaryotic ribosomes, found in plants, animals, fungi, and protists, are larger. Together, they weigh 80S. This eukaryotic ribosome is composed of a 60S large subunit and a 40S small subunit. In addition, in prokaryotic cells, ribosomes float freely in the cytoplasm. However, in eukaryotic cells, ribosomes can either be free in the cytoplasm or attached to the rough endoplasmic reticulum, where they synthesize proteins for secretion or use in membranes. Imagine stepping inside a bustling factory within a cell, where raw materials are processed, refined, and transported with precision. This factory is the endoplasmic reticulum. The endoplasmic reticulum is a vast, membrane-bound network found in all eukaryotic cells. It stretches from the nuclear envelope and weaves through the cytoplasm, connecting and communicating with other organelles like the Golgi apparatus. But endoplasmic reticulum factory has two specialized sections, each with its own unique function. First, we have the rough endoplasmic reticulum. This rough endoplasmic reticulum is located right next to the nucleus where it received mRNA. It's like a production assembly line that has flattened sacs called cisternae. These sacs are covered with ribosomes. The ribosomes located on rough endoplasmic reticulum are responsible for synthesizing proteins, which are then modified and packaged within the rough endoplasmic reticulum before being sent to their destinations, as we will see later. Further down the assembly line is the smooth endoplasmic reticulum. Unlike the rough part, the structure of smooth endoplasmic reticulum is composed of tubules. Also, it has a smooth surface, as it lacks ribosomes. Instead, the smooth endoplasmic reticulum specializes in lipid metabolism, detoxifying harmful substances, and storing calcium, making it especially important in liver and muscle cells. Therefore, we can recognize now that inside the endoplasmic reticulum, two major types of biomolecules, the proteins and lipids, are synthesized, processed, and prepared for transport. In the rough endoplasmic reticulum, ribosomes act like miniature assembly lines, synthesizing proteins, which are then inserted into the lumen. Here in the lumen, these proteins undergo folding and post-translational modifications to enhance stability and function. Once ready, these proteins are packaged into vesicles that bud off and travel to the Golgi apparatus for further processing, sorting, and final modifications before being shipped to their destination. Meanwhile, the phospholipids, cholesterol, and triglycerides that are synthesized in the smooth endoplasmic reticulum has two fates. First, these lipids can be integrated into the endoplasmic reticulum membrane. Second, like proteins, lipids synthesized in the smooth endoplasmic reticulum are packed into transport vesicles and sent to the Golgi apparatus for further modification before being directed to cell membranes and mitochondria, or secreted as hormones.